Hi, my name is George. I'm from the Plant Accelerator, the Adelaide node of the APPF. Today I'm going to show you our exciting new web tool, Zegami. Let's start off with where to find it. Zegami can be found at zegami.plantphenomics.org.au or by following the link on the APPF website. Now you know where it is, let's talk about what it does. Zegami allows users to filter, sort and chart data with the unique feature of being able to group that data with the corresponding images. As our example, we're going to look at experiment 251. Here, Dr. Trevor Garnett is investigating the nitrogen use efficiency and water use efficiency in 10 varieties of wheat. When you first navigate to Zegami, you are greeted with a list of all publicly available datasets. Currently there are just a few, with more experiments being uploaded every month. Now it's time for a demo. Clicking on experiment 251 will open it up in a new window. Now the initial load can take a little while, but once the dataset has been downloaded, it's quite responsive as it only loads higher resolution images as you look at them. Now that it's loaded, let's take a quick look at the interface. Each of these small squares is an image of a wheat plant. You can click on one to zoom in. You can also use the arrow keys to move left and right between them. You can zoom out, allowing you to view plants side by side, or zoom in to see some finer detail. You will notice that there are two panels on either side of the images. The panel on the right is the metadata panel. This will show you all of the information about that specific image, from the plant's ID, genotype and treatment information, through to measured information such as watering data and the analysed image data. The panel on the left is the filter panel. This allows you to explore the entire collection and restrict your selection. You can apply multiple filters at once, and you will see the collection resize on the fly. For example, maybe we only want to see the well-watered, low nitrogen, cultivar 1 plants. There are more ways to display the data, including the sort by and group by fields at the top. For example, we could sort by the snapshot timestamp. Group by works really well in the graph view. For example, we could group by replicate to separate the plants into individuals. The last of the major views is the table view. This separates the data from the images again, but allows you to quickly and easily sort and explore the full dataset. Let's take a closer look at some of the more powerful features of the filters. You notice that each filter, or dimension, has a type, either numerical or a list. For example, genotype IDs are a list of the varieties of the wheat, whereas projected shoot area is a numerical measure which correlates to biomass, and thus is expressed as a range. Zegami allows us to create box plots for non-numerical values and scatter plots for numerical ones. Let's look at the box plot first. Suppose we wanted to get a view of how well all the plants were doing by genotype on the final day. First, we should restrict the time after planting to show us just day 76. Next, we can go to Genotype ID, select the box plot drop-down, and choose Projected Shoot Area. This will draw us a small, quick graph in the side panel. Let's blow it up by pressing Expand. Now we can see that cultivar 1 has what appears to be an outlier. Let's try another example with the scatter plots. Suppose we wanted to know a bit more about that outlier in context. Let's restrict down to cultivar 1, select time after planting, scatter plot, and again pick the projected shoot area. Let's expand it up again, and we can see that indeed one plant appears to have deviated quite strongly. The scatter plot allows us to graph a third and fourth dimension. Let's select Series and choose Treatment 1. This will separate the points into different colours based on the level of nitrogen. If we then choose Trellis and Treatment 2, The plants are then separated into which plants were treated identically. To take a closer look at the images, let's circle the points. Watch as Zegami applies the filter in the background. So now that you've seen what Zegami is capable of, let's talk about where to go from here. If you're interested in Zegami or want to know more about how Zegami can help you make sense of your data collections, visit the website at zegami.com or email the Zegami team, info@zegami.com. As we mentioned earlier, more experiments are being uploaded and processed all the time, and you'll be able to see announcements on our blog. Well, Zegami is designed to help us share our data with all researchers. If you're a customer of the Plant Accelerator, you can gain access to your experiment prior to a public release by contacting us. You'll then be able to log into Zegami and see your collections alongside the public ones. Authentication is managed through the Australian Access Federation, so you will log in using the credentials from your home institution or visitor accounts available through the University of Adelaide. Thank you for watching, and if you have any comments or queries, please get in contact.